Hello, welcome back. In this lesson, I'm going to talk about the way our body reacts to a virus infection. First of all, I need to tell you how viruses enter our body. They usually enter our body through our skin or through our mucous membranes. They obviously enter more easily by our mucous membranes because they are free from the normal keratin superficial layer that you find on the surface of your skin. So essentially, viruses enter our body through our respiratory system, our digestive system, and our genitourinary tract. And have you ever asked yourself how your body reacts to a virus infection? Well, you already know that you have an immune system. So, the immune system is made of two parts. The first one is called the primary innate immune system, and the second one, the secondary immune system, or better, immune response. The primary response is local, is really fast and aspecific, and it's also free from any immune memory. While the secondary response is wider, is systemic and more efficient, and it's also characterized by having an immune memory. It means that whenever your body meets the same antigen for the second time, it's able to respond in a faster and more effective way. The primary immune response is made of our anatomic barriers, chemicals barriers like pH, temperature, and others, while the secondary immune response is made of some immune cells like natural killer cells and macrophages. The fever is one of the most important primary responses because it prevents the virus from replicating. The virus is not able to replicate under high temperatures. Then your high body temperature induces your liver to confiscate zinc, iron, and other essential molecules for the virus replication. Then high temperatures accelerate all the metabolic reactions. It accelerates chemotaxis, it accelerates all the cell repair mechanisms. So don't take antipyretics whenever you got temperature, because you prevent your body from fighting against the virus. Consider that temperature is toxic for your nervous system only when it's over 41.5 degrees. Another important primary response system is made of interferon. The Interfer system was found in 1957, and it's made of about 30 proteins, which aim is to block, to stop cell proliferation, virus replication. These proteins are inducible cellular proteins, and they act with a hormone-like activity. So these proteins bind some receptors and they activate some signal pathways which end by activating the transcription of about 200 of genes. These proteins have a cell-modulating activity and also an antiviral activity. They are able to induce an antiviral state from 24 to 48 hours. These proteins are coded by 26 genes. These genes are located into the chromosome number 9 and the chromosome number 12. So let's talk about the interferon molecules in a more specific way. First of all, we have the beta interferon, which is produced by epithelial and fibroblastic cells. It's a glycoprotein, a type 1 monomer. There's only one allelic variant. It's non-diffusible, so its aim is to create a sort of antiviral barrier that borders somehow the local infection. Beta interferon is produced after the cell meets a viral genome. It has a strongly antiviral activity and only partially a cell-modulating activity. Then we have alpha interferon. It's produced by leukocytes. There are 15 allelic variants. It's a protein, it's a monomer type 1. It's diffusible, so it communicates the infection at a distance. It's produced after the meeting from the cell membrane and the virus surface. Its activity is strongly antiviral and partially cell-modulating. And finally, we have the most important interferon, which is the gamma interferon, or better called immune interferon, which is produced by activated T lymphocytes. It's a glycoprotein, a dimer type 2. It's non-diffusible, and its activity is highly cell-modulating and only partially antiviral. Actually, there are other interferons like Lambda interferon, also called interleukin number 28A, 28B and 29. There are 3 subtypes of Lambda interleukins. So, which are the specific functions of this system? First of all, the system prevents the virus replication, the cell-infected replication, the expansion of the infection, and it also strengthens the immune system activity. The interferon system also activates the oligoadenylato synthetase pathway that induces the activation of a ribonuclease that essentially cuts the virus RNAs. Its half-life is really short, so it doesn't have the time to damage any cellular structures. The system also induces the activation of a protein kinase R. Protein kinase R phosphorylates and inactivates one important factor for the protein synthesis. This factor is called EIF2. Then, this protein also phosphorylates the histones, inducing the cell from blocking the cell growth. The interferon system also induces the synthesis of protein MX, which inhibit the replication of some specific viruses like the flu virus. It also induces the transcription of some proteins that block the replication of specific parasites, and finally it modulates the glycotransferase proteins that are enzymes that modify the membrane fluidity and the maturation of the viruses. Sometimes the interferon is also used in a pharmacological way, especially when there are dangerous virus infections. So it's inoculated after having been associated with a long molecule called polyethylene glycol. And it's also inoculated with other drugs. Which are the side effects of the interferon? Well, first of all, the flu symptoms, then headache, nausea, vomit, diarrhea. anxiety, depression, irritability, hematological abnormalities, autoimmune disorders, and many others. I'm sure you already know them because every time you got temperature, every time you've got the flu, you experience these symptoms. So I'll see you in the next lesson when we'll be talking about drugs. See you, bye.